Hi, this lecture is to accompanying the Chapter 11 in OpenStack's Microbiology. Contains tons of details on the central dogma and DNA replication, things like that. So let's get into it. It's long. We'll try and summarize, give you a summary overview. All right, so we're going to talk about from DNA to protein. This is microbial genetics. We're going to go over the structure of DNA, how DNA is replicated, and then the production of proteins. Tons and tons of details in here. This is stuff that took years and years to figure out, and many multiple Nobel Prizes. Lots of people worked on it. Okay, so recall that the Watson and Crick model for DNA structure, it was chains of nucleotides, that's sugars, bases, and phosphates linked together. And this is a double helix molecule where the backbone are sugars and phosphates covalently linked. And then the strands are held together by weak hydrogen bonds that are between the bases. Adenine forms two of these with thymine, and cytosine forms three hydrogen bonds with guanine. And the information here is stored in the sequence of bases. And those sequence of bases are used to make an RNA copy of a portion of the DNA molecule, which is then converted to a sequence of amino acids or a protein. This is the central dogma. DNA makes RNA, which is used to make protein. And this is the central kind of mechanism of inheritance, where the sequence of bases, the genotype that's coded in the protein, is used to produce a physical trait. And this is precisely controllable enough in that the environmental conditions can allow organisms to turn genes on and off. So different environmental conditions can produce different phenotypes. Um, Almost immediately when Watson and Crick discovered the structure of DNA, they recognized that this explained a lot of the mysterious, kind of miraculous precision of living organisms and the amount of traits. That the replication of DNA was easily explained then. The two strands could be gently pulled apart by separating the hydrogen bonds, and then nucleotides could be put together to match the old strands. And so this is called semi-conservative replication. So that to replicate a DNA molecule, you pull it apart, and the old strands are used as templates or a model to build the new strand. So this is called semi-conservative replication because the new molecules of DNA has a portion of the old molecule. Okay, it's again here, there's tons of details that were figured out in how this is done. The main enzymes involved are kind of shown in this picture here. And for us, the key ones are DNA polymerase, which is the one that adds and binds together the nucleotides. It makes sure the nucleotides match the existing strand, and then it links them together. Other enzymes do things like they started off, there's a primerase. They separate and unwind the strands. DNA gyrase and helicase do that. And then because the enzymes don't work continuously, the primer has to be taken off and the strands have to be healed. And there's DNA ligase to do this. And so here's a list of those enzymes that are there. For us, the ones really to know is DNA polymerase, which is the one that actually forms the bonds between the nucleotides, and it can prevent mismatches, so it does repair. And then the DNA ligase. And the reason we'll know those is because they're used in genetic engineering. And so the old strand serves as a template, and DNA polymerase makes sure that thymine is across from adenine, and adenine is across from thymine, and cytosine is across from guanine, and vice versa. And so the enzyme doesn't have to know the sequence, it just makes sure that the fit is right. And then lots of ATP is used to build these. And so ATP is used to energize the nucleotides and form the bonds. And so this is how DNA replication occurs. Now, there are slight differences between eukaryotes and prokaryotes. In bacteria, what happens is a replication bubble occurs, and there's an origin of replication, and at the origin of replication, the strands separate, and then DNA polymerase makes a new strand, and that's how it does. So that was one of the great mysteries. It's a bunch of enzymes that work. We've isolated those enzymes. We can purify them. We can get them to work in a test tube. That's part of genetic engineering. The second big mystery was the how proteins are made, how DNA is converted to traits. And this involves two steps, transcription and translation. In transcription, the DNA strands are pulled apart again, and the DNA strand, one of the strands, serves as a template to make a messenger RNA that's complementary to that template. And again, here there's an enzyme, RNA polymerase, that does that. And I should mention these names tell you what the enzyme does. RNA polymerase makes a long molecule of RNA. A long molecule is a polymer, and so it makes RNA. DNA polymerase makes a long molecule of DNA, a polymer, so DNA polymerase. In transcription, the main enzyme knows where to sit down, and it transcribes the DNA to RNA. And this could be messenger RNAs, transfer RNAs, or ribosomal RNA. And at the beginning of each RNA that has to be made, there's what's called a promoter sequence. And this is how RNA polymerase can find that. And RNA polymerase always works in one direction. And when it finds a promoter, it'll open the strands and keep adding complementary RNA nucleotides until it reaches a particular sequence called the terminator. Okay. And so transcription involves a small separation, and it involves the enzyme. And again, we've purified the enzyme. We can get it to work in a test tube. Okay, and so that's the first step in making a protein. RNA polymerase works. And again, it does this by matching nucleotides that are made of the right bases, and it makes sure they fit exactly. And so RNA polymerase doesn't have to know the sequence, it can copy any sequence. Then the second step is called translation, because here, instead of going from DNA codons to RNA codons to another type of codon or nucleotide, we go to amino acids. And so here we use the codons are a sequence of 3 bases, and there are 64 possible 3 base sequences that are there, and they code for 20 amino acids. Several sequences code for the same amino acid, so we call the code degenerate. And then that code that's made on the messenger RNA will be read by transfer RNAs, which carry complementary codons, and on one end, and on the other end, they carry amino acids. Here there's start codons and stop codons. And so this shows you the possible codons in RNA bases. Uracil has replaced thymine here, so if you have UUU, that is converted to an amino acid called phenylalanine, so they just abbreviate it there. And then also UUC codes for phenylalanine. If you get UUA, that codes for leucine. And so those are there, and it shows all the possible 64 codons there. Some of the codons are code for the end of the protein. Those are stop codons. And there is one that codes for the start. That's AUG. It's referred to as the start codon. I don't know why this is in here. It's the same thing, just a different version of it. So, in the process of translation, every messenger RNA will have near its 5' end, it'll have an AUG codon. Okay, that start codon is recognized by the subunits of the ribosome which bind to that sequence, and also attracted to that will be a transfer RNA. And so the transfer RNA has a complementary codon on one end and an amino acid on the other. Okay, and when that start codon is recognized and the ribosome binds to it, then the tRNA binds exactly to it. And then the second codon can be any codon, and another tRNA will come in and it will have an anticodon that's complementary, and it'll bring the second amino acid. When it does that, the ribosome will form a peptide bond, which is a covalent bond between the amino acids. And so it'll link them together so that they are starting to form the peptide chain. Then the ribosome will move down slightly, and a new transfer RNA will bring a new amino acid in there. And the empty transfer RNA that was in that carried the methionine there will move to the E site where it will be ejected. And so we'll have the ribosome starting to move down the messenger RNA. Transfer RNAs will bring the appropriate amino acids, and the ribosome will link them together. And it'll eject the empty transfer RNAs, which will then go and get refilled. And so it just chugs along like a train down the track. And as it does so, the ribosome is linking these amino acids together. And so you get this growing polypeptide chain, and it'll keep going until it gets to a stop codon. At the stop codon, there is no transfer RNA. There's a protein factor, it's called a release factor, that comes, and when that release factor sits on that stop codon, the ribosome will dissociate, and the codons will separate, or the transfer RNAs will go away, and the protein can then fold. It'll be released by the ribosome, and it can fold up. In prokaryotes, this process occurs all in the same compartment. Okay, in the cytoplasm of the cell, you have the chromosome, and you have the ribosomes, and you have the enzymes. And so as a messenger RNA is being made, ribosomes will attach to it, and they'll start cranking out the proteins. And so they'll do that. And often prokaryotes, all the genes that are involved in similar processes are located together. And these processes can be mapped onto the chromosome. And this allows them to activate it, and it's a compact arrangement. It's kind of different in the eukaryotes. We have ribosomes, we have RNA polymerase, but we modify our ribosomes more, we target it, we make things one at a time. So this is very complicated, but you should know what a ribosome does, what transfer RNAs do. There's stop codons. You should know what a promoter is. There's a lot of terminology for this. And so look at this. This is the central dogma. It's the central thing all cells do. So very, very important. And in particular, this second page of terms is really important. Promoters, RNA polymerase, translation, tRNA, mRNA. These are really, really important things. Important in cell biology and important in genetics. So we're going to stop there. I'll talk about the rest of the stuff in lecture. This is a big chapter with lots of details. Focus on the outline of the process. Be able to describe things in outline. And if you have questions, let me know. All right. See you later.